Okay, great. So first of all, I want to thank Nivedita and the other organizer like Rachel and Marcin for keeping this initiative alive. It's extremely useful. And I've seen several of the videos and I participated to some of the meetings on Zoom, and I'm really honored to be here. So today I will discuss a couple of unpublished projects on which we're working now. So I'm very much looking forward for your feedback as well. So actually to set the frame, I would like to use this picture from a review that we wrote and has been just accepted on annual reviews, where you see the typical timescales that are relevant for RNA dynamics and the computational methods that you can use to study them. In particular, in this talk, I will discuss about applications that we have done using atomistic molecular dynamics, which is in this range here. So as you see from here, to access, so the timescales that you can access with MD are up to, say, microseconds to milliseconds, depending on the system size. And you really need to use enhanced sampling if you want to sample better. On the other hand, I will also show some application done with secondary structure prediction methods. So here you can access, so you have several approximations, but you can access larger systems and also go to virtually like infinite long time scale. So you can sample basically at equilibrium. Okay, so these are computational methods. So they are affected by error, similar to experiments actually. And you can have both precision problems and accuracy problems. So when I say precision problems, what I mean is that basically your calculation is not fully reproducible, or maybe if someone else runs it in the same condition, will obtain a different result, or maybe if you run it longer, you obtain a different result. And in order to tackle this, you have to actually run simulations either for very long time scale or take advantage of enhanced sampling methods. And I will show something actually in the first project that I'm going to share with you about developing enhanced sampling methods exactly to accelerate magnesium RNA binding. These type of techniques are not needed when you do secondary structure prediction, because in that case, you can basically use tricks related to dynamic programming that allow you to virtually enumerate the entire ensemble. So at the same time, you might have problems. So even if you have if you achieve infinite precision, you could have accuracy issues because your prediction could not match experiment. And the only way to try to fix, to improve accuracy, I would say, is to integrate methods in the pot. So the very base level of integrative methods, the 0 level is just validation against experiments. So try to do some prediction that can be directly validated. And today we show validation against cryo-EM experiments. Or you can also try to do integration. And here I will show integration of DMS data into the ensemble prediction. Okay, so this is my agenda for today. The first project, as I already mentioned, is about RNA-magnesium interaction. Is related to our participation in the one of the CASP challenges. And here I will show you 3D dynamics with molecular simulations, enhanced sampling and validation against cryo-EM. Whereas in the second part, I will switch to something actually very different, which is our attempt to build a physical model for to interpret chemical probing experiments in the context of secondary structure and prediction of individual structure and of ensembles, integrating DMS data. Okay, so the first project is about binding of metal ions. So I think I don't have to explain why metal ion binding is important for RNA. Let me just remind you that magnesium is actually very important in some cases for catalysis. So it has been shown to be required for some ribozyme to be functional. But more generally, magnesium is very good at screening basically phosphate repulsion. So it's typically needed to have compact three-dimensional structures. You can have magnesium that is directly bound to RNA. So it becomes typically pentahydrated and directly bound to a phosphate, or you can have magnesium that is indirectly bound. So it's hexa-coordinated with the like outer binding structure, or you can even have diffuse magnesium that just floats around and screen electrostatic interaction with some average effect. So the tool that we will use for this first application is MD simulations. So I hope most of the people are at least familiar with the idea. Let me just remind you, this is atomistic and dissimulations. So even though in these movies, typically I represent only the solute, water is actually explicitly modeled there. Systems are described at the atomistic detail, so all the atoms are there. The interactions are parameterized, motivated on the chemistry, and we do not include polarization effects or chemical reactivity. So this is a standard pairwise additive force field. In particular, we use the Amber force field. With this type of settings, we can reach timescales of approximately hundreds of nanoseconds per day or microseconds, maybe depending on system size. Okay, so let's go to the first application, which actually, so we jumped into it because we received this email from Rachel and other people that were trying to set up a new challenge for CASP, where the idea was to basically, given this previously published structure of a ribosome, and potentially given a previously published cryo-EM map, the task was to predict the solvation shell, which in this context meant you had to submit RNA structure and also a layer up to 10 angstrom of water and ions around. And this would have been validated against the cryo-EM density maps that were measured but unpublished at the time, that were actually published by Rachel later this year in a Nature paper that I recommend you to read. So our background is MD simulations, RNA. We already worked in RNA-magnesium interaction. We were already working at that time in MD cryo-EM integration. And so we also had a running grant exactly on this topic. So we said, why not? This is actually something that we have to try to do. So I went to my collaborators, Olivier Languin-Cattoën and Elisa Posani, and we made a plan to be able to submit our prediction before August the 6th. That if there are Italians in the audience, you know it's not the best deadline you could set for something like this. Anyway, we were all on holidays actually at that time. I was just connecting in the morning to continue the simulations, check if they were running, and just to be able to collect the zip file to submit on the very last day. Okay, so this is the system that we have studied. So these are the simulation details. Maybe the only relevant thing is to notice like the size, it's around 400 nucleotides, so it's like a medium-sized RNA. For MD simulation, I would say it's quite large. Then another important thing to notice is that the way we modeled magnesium ions was through the micro-magnesium force field developed in the group of Nadine Schwirtz a few years ago. This force field was developed to match exactly the binding free energy of a magnesium on a phosphate. So basically the affinity, the likelihood that the magnesium binds on the phosphate is compatible with experiment. And this I think is very important for a challenge like this one because you want the correct number of magnesium ions to bind. If you try with a standard amber force field, you will find a much higher affinity, which implies many more magnesium ions bound. In addition, this force field was parameterized to have the correct barrier here, which is good for the physics, let's say, but it's bad for the exercise of doing MD simulations because this barrier is very high, and so it will take a lot of time to sample direct binding events. And in fact, if you run MD simulations of the system, so this is actually a simulation starting from the previously published PDB. So there are some magnesium ions that are directly bound here in pink, that were directly bound at the beginning of the simulation. And then there are many other magnesium ions which are shown around that just float around and can never bind directly, basically, because they have no chance to cross this barrier. So because of this, Olivier developed a kind of advanced enhanced sampling method where the idea is to add ad hoc energy to a term to the energy, which takes into account the distance between each magnesium and the closest phosphate, and also the coordination number with water of that magnesium ion. So in a nutshell, the idea is to stabilize the transition state associated to binding of magnesium to phosphate. In this way, the magnesium ions that you have in your simulation will become in some way more reactive, let's say, or more capable to bind and unbind easily to phosphates. And this can be easily seen in a sample simulation with a dinucleotide. However, there is a problem here that we are cheating, we are modifying the energy function. So in fact, the result is affected by this change. It's not just the timescale that changes. Also, the distribution that you explore will be different. So there is technically a way to correct for this with a procedure that is called reweighting, which explained in a maybe naive way is like, our magnesium ions, they are encouraged to spend time on the transition state. So whenever there is one magnesium ion on a transition state, we have to remove that snapshot because it's not compatible with the physics. And the problem here, when you have a lot of phosphates and a lot of magnesium ions, is that you obtain something that I like to call the group picture effect. So, and Olivier had the idea of explaining this with this very famous picture. So if you have participated to conference where you take group pictures, you probably know that the probability to have at least one person with blinking eyes in the picture is proportional to the number of participants. And in fact, in this case, Pauli was caught with blinking eyes, as you can see in the picture. Okay, so that means that if you have a lot of magnesium ions and a lot of phosphate, at least one of them will always be in that situation, and you will basically discard the entire trajectory with reweighting. So you have to do something more complicated. So based on the ideas published in previous papers, we use the replica exchange procedure where we have multiple replicas. In some of them, we don't do any trick. We just simulate with the normal force field. In some other ones, we apply fully this trick. So we stabilize the transition states. And then through replica exchanges, the information percolates up and down. And at the end, we only analyze the replicas where the trick was not applied. So in these replicas, basically, the magnesium ions are always in the correct setting. They are never like forced to be on the transition state. So this allows us to generate quickly an ensemble which is unbiased. Okay, so if we run this for some time, so in this case, it was like 10 days of simulation, 160 nanoseconds per replica. This was really the preliminary implementation that Olivier did just for us to be able to submit in time. And in fact, there's nothing special in this simulation length. It's exactly what we were able to run until the morning of the very last day. But as you can see here, so at the end of the simulation, you have many additional magnesium ions that are bound, that are predicted. So they are directly bound to phosphates, and they were not bound at the beginning of the simulation. So, of course, this is a prediction. It could be good, it could be wrong. The way to assess this, okay, sorry, first, we can try to quantify the amount of enhanced sampling that we have here. So, if you analyze the conventional MD, so without this trick, you can look at the concentration of ions in the bulk. So these are the number of ions that are, let's say, far from RNA in the simulation box. And you can see that they equilibrate to a value that corresponds to the horizontal dashed lines. So we carefully tuned the setup so that the concentration of ions in the bulk match the experiment. However, the number of bound ions that you see here is basically constant, and this constant around 20 is exactly equal to the number of ions that were bound in the initial conformation. When we do this replica exchange, we equilibrate more or less to the same bulk concentration, so it's consistent. But on the other hand, we see many more binding events, consistent with the movie that I was showing before, that shows you that, yeah, you can predict a lot of new binding sites. Now we have to check if these new binding sites are correct. So what we did, we submitted our ensembles, and these were evaluated by Rachel. And actually, these are from the slide that Rachel used at the CASP conference. And you can see very nicely that both our submissions were ranked like first and second. Very good. Actually, this meant that I was able to participate to the conference. That was super interesting. And but this was just preliminary analysis made by Rachel. And after that, she improved a lot the analysis. We also contributed to the analysis to some extent. And these are the final cross-correlation. So here, this is the higher the better. So it's the cross-correlation with experiment that are in a paper that has been accepted just a few days ago. Was in the participation of most of the submitters here. And you can see here that basically you have a very high cross-correlation if you look at the cryo-EM map between two independent experiments. That tells you that we are not validating against noise. So the experiment is actually very reproducible. Then you have a very big drop, and you have the cross correlation between simulations and experiment, which means that there is a lot better that you could do. And you see that the first three submissions in this ranking are our two submissions and a submission by Simon Poblete. I'm very proud actually, because Simon Poblete did his postdoc in my group many years ago. So actually, this is like in some direct or indirect way, we had the first, second, and third place in this figure. And what's also very interesting is that these three submissions are better in this ranking than the baseline, which is MD simulation that were performed and published in this paper, that were done using the cryo-EM data to place the ions at the beginning of the simulation. So in fact, with the prediction, we were able to beat the baseline. Okay, to summarize this part, so we did it very well at CASP. We developed an ad hoc enhanced sampling method to sample quickly magnesium binding events. I'm not sure this is the reason why we did well. I think it's more likely because we used the right force field, and probably we did the setup very carefully. But still, I think it's very useful to have a technique that allows you to sample quickly and to kind of decrease the potential precision issues. So after our submission, Olivier worked a lot to improve the method that he developed, and now he has a much improved implementation that allows also to sample to predict higher density sites where you have magnesium bound to 2 or even 3 phosphates. And what we are trying to do now, and I didn't present, is like systematic comparison of the generated ensembles with these cryo-EM maps. Okay, good. I will now switch to the second part. This is quite different topic, quite different type of methodology also. So here, for this part, we are using secondary structure prediction methods. So here the idea is that the energy associated to a given RNA conformation, which is like a list of base pairs, is just a sum of thermodynamic parameters estimated from basically pairs of consecutive base pairs. This is a very coarse representation, so you really describe RNA molecules as collection of helices, and by construction you cannot include pseudoknots. But the really nice thing is that, as I mentioned already at the beginning, by living in this very simplified world, you can exploit dynamic programming, and you can virtually enumerate all secondary structures that are possible, even for systems up to a few thousand nucleotides. So you have no sampling problem at all. And in a few seconds or minutes, depending on the system size, you can basically enumerate the full ensemble. Of course, these methods, they are not perfect. They are parameterized in some way, and so there are many limitations. So the standard approach in the literature now is not to just use them alone, but to combine them with the chemical probing experiment, like SHAPE or DMS. So in this type of experiments, what you do is you use small reactive molecules that typically can bind RNA nucleotides in positions which are unpaired or flexible. What you do then, you use reverse transcription to produce complementary DNA that are either truncated at the position where you have the chemical modification, or they incorporate a mutation. Then by counting the number of truncated DNAs, or the number of mutations, you can estimate basically the probability to have a modification in that position. The standard way to then include this experimental data in secondary structure prediction is to use pseudo-free energies, like a penalty. You penalize the formation of a base pair in nucleotides that display a high reactivity, which actually means a high number of adduction in that position. Okay, so this is a very standard approach, but there are two things that I don't like very much of this idea. So one is that these two parameters, M and B, have no physical interpretation at all. At the beginning, Naive said, so this M should be KBT. No, it's not. And it just fitted on experimental data. And I think there is no clear interpretation. And the second thing that I don't like is that In this way, you are basically overcorrecting your prediction when the prediction is already correct. Also in cases where it's already correct. So if a nucleotide is already like 90% paired, and maybe this is compatible with experiment, you will make it even for a higher fraction of your ensemble paired. So what we try to do is to develop a model where we can try to incorporate the physics of the interaction in a meaningful way. So the idea is to take a set of molecules, like these are different RNA molecules. The idea is that once we learn how the physics of the interaction with the chemical probe works, this is transferable across different molecules. We can measure them at different concentration of the chemical probe, and typically you would have a control channel where you have no chemical probe and at least one experiment with one concentration of the chemical probe, but you can easily and straightforwardly combine chemical probing done at different reagent concentrations. And then we have a model where we have like the probability of binding, in this case, a DMS molecule on an unpaired nucleotide, the probability of binding on a paired nucleotide, the probability that this transforms into an adduction. Also, we can model the probability that this adduction induces a mutation, for instance, and also the error rate in the sequencing. So we can model all the steps of the experiment, and this results in a total of something like 15 parameters that are all interpretable, that we can fit by generating an initial hypothesis for the RNA ensemble and fitting these parameters on the measured experimental data. And once we have these parameters, we can try to abandon the idea that the original ensemble predicted with Vienna, with the thermodynamic model was right, and try to further optimize the ensemble to obtain the ensemble that best match the experiment. So let's see this in action. So we made some tests on a set of structured RNAs from a paper by the group of Redmond Smith. They were published a few years ago. Here there are like 5 different systems. These are structured RNAs, so the structure is known. And but there are these very nice DMS data at multiple concentrations, all in the same settings. And here the normalized loss basically tells me how much my model is in agreement with the experiment. So the lower, the better. We made a sort of train test splitting where we use some of these systems to train the physical parameters and some to test them. And you see that the loss actually is very similar for a given system if it's used either in training or in test. There is a very, very minor difference. So that's to say that the physical parameter that you learn from one system can be basically transferred to the other system. Good. So this is just this is fitting the physical parameters. Then we can look at what happens when we try to change the structural ensemble of RNA. So in this figure, I show the same quantity, which is this normalized loss. So the lower, the better. And this point here corresponds to the ensemble that we obtain using Vienna to generate like base pairing probability. Then what we try to do is some experiment. Let's say we first try to shrink our ensemble to a single structure. So we say, what if there was only the minimum free energy structure? So we add the soft constraints so that we convert gradually this ensemble to the minimum free energy structure here. And you see that the agreement with experiment is getting worse. So that's to say that if you use the single MFE from Vienna, you get worse agreement with experiment. This makes sense. What's striking is that if we do the same exercise by shrinking to a single structure, it is actually the correct experimental structure that is known for these systems. We see a non-monotonic behavior here. So at the beginning, informing the model with the known structure, with the correct structure, makes it better. But at the end, it makes it worse. So that means that a single structure, even if it's the correct one, is not as good as an ensemble to explain experiment. And in fact, we can push this more and explore the space of all the possible ensembles. And in this case, by construction, we are minimizing this loss. So we are able to build ensembles with a lower loss, which is significantly lower. It's down here. Okay, how do these optimized ensembles look like? So again, we started from the Vienna ensemble, and then we modified to maximize the agreement with the DMS experiment. So we didn't use the reference structure information in this, but we can still compare our base pairing probability matrix to the contact matrix that we have in the known structure. So you see here the difference between the two matrices from the Vienna ensemble. If you do the Vienna MFE, we are going farther. So the prediction, the assumption of using a single structure in Vienna from the MFE makes the prediction worse. If we use our DMS data, actually we get better. So that's reassuring. So using DMS, we go towards the correct experimental structure. And you can also look at the details of the arc plots to see exactly which are the helices that you are recovering that were not correctly predicted by the original Vienna model. Okay, this is basically local dynamics. What I'm saying is that by allowing these local fluctuations, I can better describe the observed DMS reactivity. But the natural application of this should be to look for bistable RNAs where you have two completely different structures that compete. And in fact, the standard way to analyze these systems is to use methods like DREAM, DRACO, or DENSE that they use correlated data or single molecule data, sometimes people call them, which basically means individual reads. So you look when there are two DMS modifications that are correlated between each other. So in order to use this method, you need a huge number of reads because to have statistical significance in this in data, you need many occurrences. So we try to use our method in this type of application. And let me remind you, we do not use these correlations. We just use the average modification rates. So this is a designed bistable RNA. It just, you see from the arc plot, it's just two competing hairpins. One is shown here and the other one is shown here. And what we do is we construct two ensembles with either one or the other hairpin. We generate synthetic reactivity data. We mix them in a known fraction, let's say 80% and 20%. And then we give to our method the mixture and see if the method can predict the correct population. And as you can see from this inset, basically the population that we predict for the two helices is almost identical to the, in this case, known population with which we mixed the two datasets. Okay, this is with the synthetic data, but actually these are realistic and real sequences, including primers, and the experiment is now in the pipeline of our collaborator, Redmond. So we try to also apply this to stretch a bit the model and apply it outside of its comfort zone. So as I said, what you expect here is to have experiments that are done in the same conditions. We try to see what happens if you apply to experiment done in different conditions. Like we try to fit the same physical model to experiment at two different temperatures. These are data from a CSPA 5' UTR RNA thermoswitch. And what we can see is that by using this approach, so even if we force the interaction, the parameters determining the interaction between RNA and DMS as the same in the two experiments, we can still recover the correct reference experimental structure, which is different at the two temperatures. And also in this case, we can perform the same exercise that I was showing before. And it's actually very similar to a test that was used to validate the DRACO method. We mix the two experimental datasets in known fraction, and we try to recover the population of the two states. And you see in this case, the agreement is not perfect, but we have actually a very good estimate of the population by analyzing our ensembles with respect to the known mixing fraction. Okay, to summarize this second part, so we built a physics-based model for chemical probing, where we take into account the way probes interact with paired or unpaired nucleotides, and also the existence of multiple RNA conformations. And the nice thing of this idea is that you can use it to reconstruct ensemble from average mutation profiles. In principle, we know formally also how to include single molecule data. We didn't try yet in practice, but it would be interesting to see if including single read measurements could improve the result or not. So I already shown you how you can combine multiple DMS concentration. We tried to combine also multiple temperature. In principle, one could extend the model straightforwardly to combine different reagents just adding number of parameters, like shape and DMS experiments on the same molecule. And one could even try to model experiments taking at different time points, modeling the kinetics by which DMS interact with RNA. Okay, so this is just to wrap up. These are the two projects that I discussed, very different from each other. So one is atomistic MD simulation of magnesium RNA interactions validated against cryo-EM. And the other one is an approach to integrate DMS data with secondary structure prediction that can be used to build ensembles. And finally, let me acknowledge my collaborators. So the first work on the CASP challenge was done by Olivier, who is also here in the room, and also with the collaboration of Elisa, that was our she's a previous PhD student, and she was our local expert in the analysis of cryo-EM maps. And for the second project, I have to thank Giuseppe Sacco, a PhD student, and this is done in collaboration with Guido Sanguinetti at CISA and Redmond Smith at IBMC. And then let me thank funders and all of you for your attention.
All right, so questions can go in the chat, or people can raise their hands. I can get started with a question because I have one about your last slide on your second topic about the kinetics. Is there any way right now you get an ensemble, I assume with weightings of population? Is there any sort of time information in the data you get, or is it just population?
No, there is not. So maybe it's possible also to do that, but what I meant here with kinetics is the kinetics of DMS binding. Because in fact, these experiments are by construction out of equilibrium. If you wait a lot of time, you will have more modifications, and this we do not include it. It would be cool to also include RNA kinetics, but I'm not sure it would be easy to do it in this framework.
All right, Simone, do you want to unmute yourself?
Yeah, can you hear me?
Yes.
Yeah, okay, cool. Okay, so yeah, very nice talk, Giovanni. Thanks also for the reference. I had a very short question about the DMS treatment because I have read also some other papers which involve 3D structures for making the modeling. I don't know if it's exactly the same thing that you were talking about here or is something that goes beyond. So my question is mainly related to the do you also consider non-canonical base pairs into the modeling that you can do with DMS, or this is also applicable to non-canonical pairs that could be in other regions?
Yeah, very good point. No, we don't. We completely ignore the thing at this stage, and we only use the secondary structure representation. I don't know if the paper that you are referring to is the same that I have in mind. There is a very nice Bayer-Clive paper by Josh Yesselman doing a very systematic analysis of
Yes.
structural motifs, and that's the way to go. But we are not doing that, at least not yet.